Hello, in this video I show you how to determine distance between two index groups using GROMACS GMX distance tool. Specifically, this tool allows you to track down changes in relative distance between atoms in two groups. For example, if you want to determine whether or not certain complex is stable based on the distance between ligand and protein atoms, you want to calculate the average distance and the descriptive statistics, which includes variance and standard deviation. In this video I cover all types of output possible with GMX distance program. To perform distance calculations you can use a command which you can see on the screen right now, and I will also add this command to the description of this video as I usually do. Here I provide GMX distance with pre-aligned GROMACS trajectory, where the complex is centered in the box, all the water solvent molecules are removed, and periodic boundary conditions are removed as well. Here I specify static index groups for which to perform distance calculations, but you should note that you can also use dynamic selections. Let's check the output files. As you can instantly notice, two of the output files are fairly large. This XVG file contains distances between atoms in each index group. This XYZ file contains X, Y, and Z components of those distances. You usually don't need these two files in most cases. This average XYG file contains average distances between atoms of these two index groups. There is also a file with descriptive statistics, which includes variance and standard deviation of distances, and another file with probability distributions based on the frequency of each distance between the two index groups in your trajectory. By combining this data with other metrics like RMSD and hydrogen bond count, you can determine whether or not your protein-ligand complex is stable. If it is not stable, you can also use this data to determine at which time point the dissociation of your complex occurred, and you can further visualize this particular frame using programs such as VM to determine what exactly happened. Now let's plot data in these three last files. As you can see, average distance between atoms in our two index groups does not vary significantly, which clearly indicates that our complex is stable. In this figure, the probability as a function of distance between two index groups is plotted. Here, distances with higher probability peaks are the most frequent in your trajectory. On this graph, the standard deviation of individual distances of atoms in your index groups is plotted. Thank you for your time, and let me know if you have any questions.